Okay, now if we do an active process, this is going to require energy. And we're typically moving things from low concentration to high concentration. So moving it, in this case, we're inside the cell to outside the cell. But it could be opposite, okay? We could be moving against our concentration gradient, bringing something in. So it could be low out here, outside the cell, high inside the cell, but you're still moving against that concentration gradient to bring even more of whatever you already have a high concentration of into the cell. So if we're going to make these types of comparisons for this one, then we'd say that we're moving from an area of low concentration to an area of high concentration. We'd say that we're moving against a concentration gradient, or we're moving up. Okay, so instead of down, we have up, and instead of with, we have against. So active transport requires energy. Typically this is in the form of ATP. Okay. Energy is used, so basically, like, let's think of you rolling down a hill, okay? You're moving from an area of high potential energy to an area of low potential energy, okay? You're moving from, if you start rolling down that hill, what are the chances that you're automatically going to start rolling back up the hill? Probably not very good. So to roll back up that hill, you'd have to stop, which would take energy. You'd have to roll the opposite direction, which would take energy. So you're going against the natural tendency of what you want to do. The natural tendency wants to go from high to low because we're always trying to create these equilibriums of the body. So that high to low example of the hill was just a random example, but the whole thing is the idea behind this is you have to ask yourself, are you moving naturally with or down a gradient, meaning are you going from high to low concentration? If you are, you don't need energy, and that makes sense. If you jump off a diving board, you're going to fall straight down. If you roll down a hill, you're going to roll straight down. You don't need energy to do that. To jump up onto a diving board, that takes energy. To roll up a hill, that takes energy. All right. So for these active transport systems, they typically use an energy and a pump. So this, for example, this is our sodium-potassium pump that we'll get into later. And it has this little tiny active site over here. We talked about active sites when we talked about tertiary structure of proteins in a video ago. So this has a nice active site for ATP, and when ATP comes in here, then we have these sodiums can leave the cell, and we have these potassiums that can come into the cell. So this is an example of active transport processes. Uses a carrier protein, in this case our sodium-potassium pump, and it uses an energy currency, in this case ATP, to pump something, in this case ions like sodium-potassium, against their concentration gradients. Now that we're looking at these active transport processes, let's redefine what is an active transport process, what does it require, and what are the different forms. So active transport processes require energy. They may or may not, so yes or no, they may or may not use a protein to help move this thing. And we move against or up or from low to high concentration, whatever you want to say. Against the concentration gradient, up the concentration gradient, or move from an area of low concentration to high concentration. So the first one we'll do is exocytosis. So osis, this is another root that we want to work with. Osis is the process of, okay, so it's the condition of. Cyto is cell. Exo means outside. So this is the process of outside the cell, or the process of moving something outside the cell. So if we have a cell, and our plasma membrane is right there, cell and plasma membrane, we want something to leave the cell. So exocytosis is going to allow large molecules to leave the inside of the cell, and it's going to use these things called vesicles. So here's a vesicle, and inside this vesicle is the thing, compound A, that I want to leave the cell. Compound A is too big just to move to the cytoplasm and across the plasma membrane through diffusion or anything, so we're using this vesicle. A vesicle is just another name for ball of phospholipids. Ball of phospholipid bilayer. So if you really look at a vesicle, here's a vesicle, you have a phospholipid like this, and a phospholipid like that, phospholipid like this, phospholipid like that, going all the way around, and then you have compound A in here. So again, it's I don't want to call it a mini cell because it definitely doesn't have everything that a cell needs to have, but it's this smaller encapsulation of phospholipids that hold something in there. So now let's go back to our picture. We want this to leave. So we have to use energy. We can't just let this thing go out. We have to use energy, and what this is going to allow us to do is this vesicle, which is also phospholipids, along with this plasma membrane, which is also phospholipids, they're going to fuse together to let this thing out. So if I do this in a couple stages, cell, molecule A. Now, molecule A gets close to the outside, and we're going to have this weird fusing of the vesicle. Sorry, here's the vesicle. Fusing of the vesicle with the plasma membrane, and then in the last picture, we're going to have this vesicle release its contents. So now A is on the outside of the cell, and this vesicle has just become part of the plasma membrane, and A has left the cell. So this is an active transport process, which means it uses energy to move something from a low to high concentration gradient. And in this case, when we're talking about exocytosis, we're using vesicles to accomplish this. Then you can do endocytosis. What do you think is going to happen with that? Opposite. So we have outside the cell, we have compound A, and we want to bring compound A into the cell. So we're going to have this puckering of our plasma membrane. That's going to allow compound A to move in there. And now we'll have a picture that kind of looks like this, with compound A in there. And then these parts of the plasma membrane, this end and this end, can come together, which is going to allow all of this to pinch off into the cell. So your next step is two sides of the plasma membrane fusing. So now we have our cell that's intact. We haven't lost any integrity. And now we have a vesicle inside the cell with our compound, and then that vesicle can dissolve, and now we have compound A inside the cell. So we have two different versions of this endocytosis. Remember, endocytosis, endo means within. Cytosis is this process of the cell. So it's the process of bringing something into the cell. And we have two forms. So we've got phagocytosis, and we have pinocytosis. Phagocytosis is cell eating, so this is bringing in large solids. And these are really important with immunity. So whenever we bring in things from the outside, we use these things called macrophages to bring these foreign substances. We bring the foreign substance into a macrophage, and then the macrophage can eliminate it. So that's where we'll see these phagocytes a lot, this phagocytosis action. And then pinocytosis, this is bringing in liquids. So if you bring in a liquid, let's just draw our picture, and we have this liquid that's, instead of compound A, now we have this liquid that wants to go inside the cell. But within this liquid, we have a bunch of different dissolved little solutes, right? So that's wherever we say pinocytosis, yes, it's bringing in this liquid, but it's also bringing in whatever is held within that liquid. So again, we'd have this part pinch off, and then this would move inside the cell in a vesicle, and we would have our whatever this liquid was, and whatever was being carried in that liquid. That's also shown. And then we can get rid of this vesicle, and now whatever we wanted is inside the cell. This is pinocytosis. Pino refers to liquids. Phago refers to eating. Cytosis is cell. So this is cell eating, this is cell drinking. We're bringing in liquids that happen to have solutes in them as well. So that's exo and endocytosis. The next one is receptor-mediated endocytosis. And that's whenever, instead of just having this little dip where we have compound A come in there and then this fusing, instead what we're going to do now, we're going to have our cell, and on this cell is a receptor, and whatever this molecule right here is going to fit into this receptor. Now that it's fit into this receptor, if you remember from that last thing, we said those receptors are sticky, so this whatever this molecule is isn't going anywhere. So now, instead of a vesicle forming that way, we're going to keep the receptor and we're going to form a vesicle around that instead. So this would now become inside. We'd have this vesicle with a receptor in it and whatever we're trying to carry inside. So that's receptor-mediated. This is just endocytosis, just bringing something in by itself, and then if it attaches to a receptor and it's brought in, that is called receptor-mediated endocytosis. All right, let's go into the plasma membrane specializations. Actually, I'm going to keep this video short and we'll start another one.